Good afternoon. I hope everyone is doing well this afternoon. As always, we'll give it till about 5 past or so, just to have a few more people join. And then while we're waiting, Sheng-An Yang, you're online. Can we just double check that you're able to share your screen and everything so that we can get going in your...
Yeah, sure. Can you see my slides now?
Yes.
Okay.
Perfect. Now that I've asked you to share, if you can stop sharing for a second, just while we go through our intro slides, but wanted to make sure that you had all the correct permissions to be able to do that.
Okay.
Okay, so we do have a reasonable number of people who've already joined, so I might get started with our announcements. So the main thing I wanted to update everyone on was that beginning this Friday, so on June 16th, we'll be beginning our ClinGen summer workshop series. So this is going to be at 2 p.m. and will take the place of our normal consortium calls. But over the summer, given that people will have less availability as they enjoy the nice weather, we're going to be running a series of workshop discussion panel type sessions across a series of topics that are of interest ClinGen wide. So we'll be kicking that off this Friday with a talk from Terry Klein from the Pharmacogenomics Working Group. And then the agenda for the rest of the series we'll put in the chat for you, but there'll be talks around the Genome Connect platform and also an overview of what's coming in the new ACMG ClinGen CAP sequence variant classification guidelines. Before we get into the rest of the content for the day, were there any other general updates or announcements from anyone on this call?
Would you speak near to the microphone?
Say that again, sorry.
Could you speak a little bit more closer to the microphone?
Certainly. I'm going to be stopping talking in a second, so I won't bother too much about making sure everyone can hear. Just to say that our next meeting is next Thursday. So because the meeting cycle that we go on is by the second and whatever it is, it means we have another call for this again next week. So that will be Les Biesecker from the Linkage and Segregation Working Group within ClinGen as part of the SVI working group. But our presentation for today is coming from Sheng-An Yang, who is presenting on behalf of the Hereditary Cancer GCEP. So I will hand over to Sheng-An.
Thank you. So yeah. Can you hear me clearly?
Perfect.
Okay, thank you. So I'm Sheng-An Yang, and I'm appreciate for having me today. And I'm here today. I'm here as a coordinator of the Hereditary Cancer GCEP. So today I would like to share our experience in the gene recuration, especially for the hereditary cancer genes. So today I will have 2 parts. So because in each part I will focus on a little bit different things. So you can see the right hand side. So here is the breast cancer recuration, and the left is breast cancer, and the right is about the colon and the polyposis genes. So so here, okay, so before the hereditary cancer GCEP form, there are two other cancer-related GCEP. The first is the breast and ovarian cancer GCEP, and the second one is colon cancer and the polyposis GCEP. So in 2018, each GCEP, they published their own paper focusing on the relationship between cancer genes and for breast cancer, ovarian cancer, or for the colon cancer. So these two GCEPs, they were inactive in 2022, last year. And because the hereditary cancer GCEP form, we take over their curations, and we maintain their curation. So we are recurating all of their curations done previously. Okay, so for this part, the first part, today, in today's presentation, I will focus on the recuration for some breast cancer genes. Although we did all breast cancer and the ovarian cancer genes, but here today I will just focus on some, because I will focus on how we use the contradictory case control data to refute these gene curations. And the second half, I will focus on the recuration for most, actually I should say all colon cancer polyposis genes, especially focus on lumping and splitting, disease naming, or the granular evaluation of new data. And I will also share the challenges we experience and what the solutions we try to apply to solve these challenges. So and one thing I want to mention is that every recuration are in the GCI. So okay, so now I would like to move to the part one. So part one is about the breast cancer gene recuration. So this actually, I reuse the slides the Dr. Julius Nitate. So he is a NIH fellow. So he rotated to our group, and we worked together for these breast cancer genes. And he presented this work at the 2023 ACNG meeting. Okay, so here, as I mentioned, so this is the paper published by the breast cancer GCEP in 2018. So in this paper, they published 31 gene disease curations for hereditary breast cancer. And in 2021, there are two major breast cancer case control study. So in these two studies, they focus on who discover the relationship between the pathogenic variant and hereditary breast cancer. So this paper were published by cancer risk estimates related to susceptibility. So hereafter, I will just call them carrier study. And the second paper is published by British Cancer Association Consortium, BCAC. So actually, they published to the New England Journal of Medicine back to back. So in carrier study, they evaluate 28 genes with more than 32,000 controls and cases. In BCAC, there are 34 genes evaluated, and with more than 48,000 cases and controls. So yeah, they are very nice studies. So we decide to use them to re-evaluate the gene disease relationship, especially for the breast cancer. Okay, so from this paper, we found that there are multiple breast cancer cases versus control comparisons show no difference in odds ratio for loss of function variants. So here, show you the list. There are 7 genes listed here. So you can see that in BCAG and the carrier study, the odds ratio is actually very low and did not show any significance according to the p-value. Yeah, and these 7 genes were classified as moderate to disputed. And because according to the guideline, we have to recurate this gene at least every 3 years if they are in this classification. So we decide to recurate these genes, and we lost 2 very large and very good case control studies. Okay, so here is our procedure. So so we incorporate these large case control studies, and we also include all original evidence, the evidence in original curation, and this original evidence was recurated per the new SOP. And we also did literature search for any new papers that including genetic and experimental evidence. So we put all experimental evidence and genetic evidence to the GCI. So then we have a meeting. So in the meeting, we have all members from prior breast ovarian cancer GCEP, and also the member in current hereditary cancer GCEP. And we re-evaluate this evidence and decide the final classification. Okay, so then I will give you the first example. This is XRCC2 and hereditary breast cancer. So here, show you the summarized table. So there are 11 independent case control studies, and they are all contradictory studies, including carrier study and BCAG study. And you can see here the sample size. There are 2 large sample size in BCAG and carrier studies, and also 9 additional studies with small to medium sample sizes. And all data show no statistical significance between the variant loss of function XRCC2 variant and the breast cancer. And we also score the supporting experimental evidence. Here I will show you some examples. So you can see that for biomedical function, we found the XRCC2 is important for DNA repair or DNA recombination. And because DNA repair and the recombination, they are very important for cancer formation or tumor formation. So we score them, but because we did not see the direct evidence showing SRCC2 with cancer or tumor formation, so we downgrade the score from 1 to half. And we also have a model organism that is a mouse model. So in the mouse model, they show the chromosomal instability. So, but again, because the mouse model did not develop tumors, so we also downgrade the score to half point. So in total, this curation can get 1.5 points. So 1.5 points, according to the default setting, that will be limited. But we put the case control, especially conflicting case control study and the supporting experimental study together, and we found that the case control study actually is more stronger and with higher quality. So we are able to use this case control study, the genetic evidence to eventually refute this gene disease relationship. Okay, and then I would like to give you the second example, that is MSH2 and breast cancer. So here I would like to point out that MSH2 was previously curated as definitive for Lynch syndrome. And because some early studies suggested that breast cancer might be enriched in Lynch syndrome patients, so we would like to curate if this relationship is true or not, according to the evidence, current evidence. Okay, so again, here we focus on case control study. So here we found 5 independent studies. All of them are, contradictory evidence. So among these 5, including carrier study and the BCAC study. And again, you can see the sample size is various. And again, there is no study shows statistical significance. And then we also scored the supporting experimental evidence, just like SRCC2. So here you can see we found the protein interaction between MSH2 and the estrogen receptor. And because estrogen receptor is very important for breast cancer, so we award a half point for this evidence. And we also found the evidence for expression. So there is no MSH2 standing in the breast cancer tissues. So we found that this one might be also helpful, especially we are looking for the breast cancer case. So we also award a half point. So in total, we got a 1 point for this. So again, 1 point actually is limited, but because of the very strong case control evidence, so we are able to refute this gene disease relationship. Okay, so here I would like to show you our curation results from 2018 to 2023, the recuration. So we can see that there is one moderate classification, the original classification, and the 3 limited and 3 disputed. So after recuration, we got 1 disputed and 6 refuted. So here actually, so I think people expect that like after recuration for the limited or especially the moderate, we expect to bump up them to moderate or strong or even definitive. But actually here are some good examples show you that with some very good contradictory evidence, actually the gene disease relationship not bump up to strong or moderate, or sorry, strong or definitive. Instead, they become refuted. So, but I think that is something very helpful because with more evidence, we are able to refute those gene disease relationship. So the clinical lab, they can consider maybe to remove these genes from their testing panel, or at least they may if they found some pathogenic variants, maybe they can pay more attention on, okay, so yeah, these might not be very important genes for certain cancers. So I think that would be very helpful for that aspect. And then, okay, so this is the last slide in this part. So here I would like to show you the new feature of the GCI. So I show you a lot of contradictory evidence, but actually before, the new feature of in the GCI, we found that it's very hard to curate these contradictory evidence. So the new feature in the GCI actually is we can use contradicts as an option, and we can select this for conflicting data, especially for case control studies. And if we select this, the entry can be displayed in the classification matrix table and in the evidence summary and in ClinGen website. So here I would like to show you how it works here. So you can see that for the supporting evidence, we can score them from 0 to 6 if that data is very good and the odds ratio is high, p-value is low, and if they have well-matched cases and control, we can award maxima a 6 point for that data. And if some like supporting data with a few concerns, we can score them from either 1 to 5. And if that is the very questionable, supporting data, we can only award 0 for this data. And now we can have the contradicts. So for the conflicting data, we can select contradicts. So that will be another category. But previously, so for the conflicting data, we only can choose 0. But that is very confusing because we don't know if that is because that is a conflicting data or the data quality is not good enough to get any score. So, but now because we can have this as an option, that will be very straightforward to see. Okay, so you can see here, I'll give you an example. So how it works here. So here I use red 50 as an example. So here you can see this is the case control evidence in the GCI page. So at the very end, so you can see, you can select status as score, review, or contradicts. So if you select contradicts, that will show you in the preview page. So you can see that here, it would be labeled as contradicts. However, for the something with score, you can see score and the number of score here. And also, let me put this to here. Okay. So here is the ClinGen website. So here you also can, people also like the reader or visitor to the ClinGen website, they also can see the scores status. So for example, this, okay, so we have several case control study here, and most of them are contradicts, but there is one with score like 1 point. So yeah, because of this new feature, we are able to deal with the contradictory data efficiently and with a very clear way to do so. So yeah, that is very helpful for these refuted or disputed gene curations. Yeah. So yeah, any questions about this? If no, maybe yeah, we can discuss in the end of this presentation. Okay, so if no major concern about the part 1, I would like to move to the part 2. So part 2, I will focus on colon cancer and polyposis gene recuration. So okay, so again, so this in 2018, the colon cancer GCEP, they curated and published 42 gene disease curation for hereditary colorectal cancer and polyposis, and we recurate all of them according to like new SOP, new evidence, and to the GCI. So I would like to show you the rationale of the recuration. So again, so the colon cancer GCEP was in activated in 2022, and the curation work is currently maintained by the hereditary GCEP. So the original gene curation was performed on gene express. That actually is an Excel spreadsheet, and on old version SOP, either version 4 or version 6. So we are working on the recuration of these genes. So all gene curations are on the new SOP, either version 8 or version 9, depends on when we curate it. And we also perform pre-curation, also known as lumping and splitting for every curation. And we also recurate this gene based on the data in original curation and the new evidence from new published. And again, so all gene curation were performed in the GCI. So here I would like to point out that, so, you know, although these curation were done at least 5 to 6 years ago, many of them are actually classified as definitive or refuted. So according to the ClinGen guideline, we don't need to recurate them just because of they are old. So we recurate them is because we want to have all data entry to the GCI, and we also want to reevaluate if there is new data, and we also want to recurate them on new SOP. So here I would like to, you know, point out that. So if you, like in your GCEP, you have something old, but if they are definitive or refuted, you don't need to recurate them, like what we did here. Okay. So first, I would like to show you the new evidence and the new SOP, this part. So as I mentioned, we recurate them, reevaluate them, the old curation with new evidence, along with old data in the original curation. So we also score the evidence with the new SOP. So we found that the point system was changed over time from like version 4 to version 8 or version 9, especially for missense variant and the case control studies. So for missense variant, they tend to receive fewer score per new SOP. So we found that we need to find more evidence to maintain the original, especially the definitive classification. And on the other hand, the new SOP, they do have more clear guidance for like how to score the case control studies. So for example, we need to consider the quality, especially if case and control are well matched, and the size of cases control as ratio, p-value of them, and we can, you know, decide a reasonable score for each case control studies. And because of the new evidence and SOP, some classification was changed. And then I would like to share our experience from, you know, Gene Express to the GCI. So I don't know, you know, how many people here they use Gene Express previously. Actually, I never used that, but I know, you know, the Gene Express actually is, you know, they document everything on a Excel spreadsheet, but because the Excel spreadsheet that, you know, so have some weakness. The first one is that it's not easy to share the data among the group. So you have to email to the person if they need. And that is also not easy to recurate if there is no new evidence. So you and the data structure is flat. So basically, you know, like all data represented as a single table, and that is a very huge table and very hard to find the data we want. And the second thing is that the Gene Express, they can only show very limited information in the ClinGen site. So we found that there is only very basic information. So for example, like a gene, disease name, the like inheritance pattern, and the final classification. And for the summaries, that can be no or very short, and also no details for genetic and or experimental evidence. But in the GCI, everything was changed. So in the GCI, it has a well-organized hierarchical data structure. So you can, like, from each paper to each genetic evidence, and you can also add every detail to you know, each category. And that is also very easy to update if new evidence become available. And also, again, so we also can add the detailed information. So except for the some basic information, now we can have well-organized summary, evidence summary. So and also thank for the standardized text, because that also guide us like what what is the important information we should add to the summary. In addition to the summary, we also can have the details of each genetic and experimental evidence. And we also can have a lumping and splitting information. So this one actually also involved in gene checker, but I found that is too much to say, so I won't share too much about the gene checker today. And also the GCEP has much better data transparency. So we found almost all data in the GCEP can be shown on the ClinGen website. So when a visitor or a reader, they visit a ClinGen website, they can see almost every detailed things about that curation. And I also want to share our experience in prioritization. So actually here I feel like it's very interesting, because how we prioritize this gene curation is based on the original classification. So the classification in 2018. So why we did this is because of the following reason. So the highest prioritization is the curation originally have definitive or strong classification. So because we recruited several new curators for re-curation work in 2022 and 2021 and 2022. So these definitive gene, they are considered as more straightforward curation for the new curators, because they have more high quality supporting evidence, and they can assign the score usually straightforward, and there are sufficient data for training. So we decide to use to recurate this definitive curation, and set them as highest priority. And then we curated the moderate, disputed, or refuted curation. Because they are more challenging curations. So sometimes the curator can find some contradictory data or not that high quality evidence. So and they need more help to help them to say, okay, so if we should treat them as contradictory or just award a fewer score. And because of this conflicting evidence, we need more discussion while assigning score. And then the lowest the curation is the originally classified as limited. So how we did for this is that we assigned multiple curations, like for example 6 curations, to a very senior curator, and ask him or her to find new evidence. And we found that actually there is very limited evidence available in not only in original curation, but also like a new evidence. So, and we decided to have them as very low priority. So we will deal with this curation after we finish all important curation. Okay, so and then I would like to have our experience, to share our experience for the pre-curation. So actually, we found the pre-curation actually is very powerful tool, especially for re-curation. So again, we performed lumping or splitting for all colorectal cancer gene recuration, and so far 9 associated disease were changed from original curation in 2016 or 2017. So as I mentioned, we found this lumping splitting is very powerful tool to determine a suitable disease entity. So just like according to the guidance, we evaluate disease entities, the model of inheritance, phenotypic variability and the molecular mechanisms. So results of the lumping or splitting, according to the results of lumping or splitting, we can either keep the original gene disease relationship, or we can lump our curation to other GCEP, or we can have new gene disease relationships. If we found we have to have a major change, we request new disease entities. We work with Mondo for this requesting. And if the disease relationship we found is very minor, we will just keep the original disease entity, but we will add additional information in the evidence summaries. So here is the example. We found we eventually lumped our curation to other GCEP. So that is SMAD forward. So this originally classified as definitive for autosomal dominant JPS. So we also found that the hemostasis thrombosis GCEP has a combined gene curation for JPS HTT. So that's like two phenotypes. It's a combined curation. And per lumping or splitting, we found that 81% of SMAD for patients with JPS, they also had HTT, HHT. And 14% of them are suspected to have HHT. So that is 95 in total. So, and we also found the JPS and HHT, they actually have the same inheritance pattern and no different in molecular mechanism. So we decide to lump our curation to the other GCEP eventually. So yeah, and we will probably retire or just archive our original curation. And then after lumping or splitting, if we found there is a major change on the disease, associated disease, we have to get a new disease name, because these disease names are usually not available in the Mondo. So here I would like to share our experience in disease naming after lumping or splitting. So here you can see, yeah, it's a post lumping or splitting task. And we found that although the guidance of disease naming is published recently, that is actually later than we request this disease entity. But the concept in this guidance and the disease naming working group actually they really help us for this disease naming. And so for disease naming, we worked with Mondo group. So I really appreciate with for Mondo and the disease naming working group. So far there are 8 new lumped disease terms in this colon cancer and the polyposis gene recuration. So here is the list. So you can see that we also worked with other VSEP for the new disease terms. Okay, so and then I would like to give you some example of the colon cancer and the polyposis recurations. So first, I would like to show you the example for POD1 and the POE. So they are the subunit of DNA polymerase, and they are involved in DNA repair. So the original curation was definitive for both of them. For the autosomal dominant, that is a very spatial polyposis type, the PPAP. After lumping or splitting, we found the germline variant of POD1 and the POE are associated with not only the polyposis, but also colorectal cancer. So we suggest to, we propose the new name to include the polyposis and the colorectal cancer for both of them. And then we recurate these 2 genes with the new name, and the new classification are still definitive. And the next example is NTHL1. So NTHL1 is a glycosylase that involved in DNA repair when DNA got damaged. So the original classification is moderate for autosomal recessive colorectal cancer and polyposis susceptibility. There is one paper published in 2019, and we rely on this paper for the lumping splitting, because this paper they report that like recessive or biallelic germline anti-HL1 mutations actually associated with a multi-tumor syndrome with a specific mutation signature, that is signature 30. So because so previously the gene disease relationship only focused on colon cancer and polyposis, not for like multiple cancer types. So you can see that many other cancer types also associated with the anti-HL1. So we decided to request a new Mondo ID, that is anti-HL1 deficiency tumor predisposition syndrome. So in this curation, we include multiple cancer and tumor phenotypes. So listed here, not only colorectal cancer, but much more. And eventually we recurate this with the new disease term, and the final classification is definitive from moderate. And the third example I would like to show you is CDH1. So CDH1 gene encodes E-cadherin, that is a cell adhesion molecule. It's very important for the maintaining the cell morphology. So original curation was limited for autosomal dominant colon cancer. But in 2022, a paper published by CDH1 group, and in the paper, they found that the germline variant in the CDH1 gene is associated with diffusive gastric cancer, and subsequently, more paper report the association of CDH1 and a very specific type of breast cancer, that is lobular breast cancer, the LBC. So in this 2022 paper, the CDH1 group, they proposed a new disease name, CDH1 related diffuse gastric and lobular breast cancer syndrome. So they combined the gastric cancer and the lobular breast cancer in one term. And because of this, we decided to lump the hereditary diffusive gastric cancer and the breast cancer curation into one curation for the new term. And we also re-evaluate the association of CDH1 for our other published. So for example, so here actually we start from the colon cancer, but we actually found that these two, gastric cancer and the breast cancer, make more sense. And we also found that there is very limited data to support the gene disease relationship between CDH1 and colon cancer or ovarian cancer. So we are consider retiring or archiving these two recurations, and we will lump the gastric cancer and the breast cancer curation into one curation. And this is in progress, and we expect the final classification will still be definitive. And the next is CTNNA1. So this is another example that we work with other VSET. So this gene encodes a alpha ketamine protein involved in the cell adhesion process. So the original curation was no known disease relationship for autosomal dominant colon cancer. So because CTNNA1 and CDH1 are involved in the same pathway, so you can see CDH1 is here and CTNNA1 is here, and they involved in the same pathway. So we also worked with CDH1 group, And eventually, the experts in that group suggest us to use ctNNA1 related diffusive gastric and lobular breast cancer syndrome as the Newton. So we recurate this gene for this disease, and recuration result is definitive for this relationship. Okay. So previously, I showed you some example that we changed the classification and the gene name. But here, after I would like to show you something we did not change the name, but we changed the classification. So the first one is RNF43. So this is the ubiquitin ligase. So this is a tumor suppressor. So originally curated as limited for this cancer syndrome. After lumping or splitting, we decided to use the same disease term, but we found that there are actually 8 papers published after the original curation. Because of this new data, we can bump up the classification from limited to definitive after recuration. So that is because RNF43 is a new gene, so there are more papers published after the original curation. So I think that is also the value of recuration, because we can have an opportunity to revisit if there are more papers, and we can have like a strong or definitive classification for that relationship. And the other is the XO1. So this one is the other the direction. So here actually the original curation is disputed for the very special type of colon cancer. So per lumping or splitting, we decided to use the same term, and we found that there are no papers published after the original curation. So we re-evaluate the evidence in the original curation. We found that the first paper to report this association, actually they did not exclude the variant of other colon cancer or polyposis associated genes. So that means that this patient have the colon cancer is probably because of other genes. It's not because of XO1. And we also found some follow-up studies. And in this study, they show that several variants observed in patients are also observed in controls with similar frequency. So that means that so probably that is just like the background mutation, or say so sorry, I shouldn't say that. That is like the normal mutation that can be seen in case and controls, and patient and healthy person. So probably the variant of this gene is not the real cause of that disease. And due to this data, we believe that this is sufficient evidence to refute this gene disease relationship. So eventually we classify this as refuted. I think I would like to skip this one because this one shows similar things as previously, because I would like to show you the summary of the our current recuration results. So here, this column show you the original classification, and this one is the new classification. So you can see for definitive, majority of them still be definitive. The only one is we decide to lump to other GCEP, and we probably will retire or archive them. So for the moderate, we have 1 to definitive and 1 to disputed, and 2 keep the same. And we have 3 limited, 1 become definitive, and 1 we found that there will be a better relationship between CDH1 and other definitive curation. So we probably will retire or archive this original curation. And we have 1 non disease relationship become definitive due to more supporting evidence, and 1 disputed to refuted, and 1 refuted still refuted. So for here, I would like to just point out the lower part. So actually, there are 3 genes. We are requesting the new Mongo ID for them. They are ATN, PowerBuild2, and Check2. So you can see that the original curation for this gene and the colon cancer is limited or moderate. So we will, after we request the new evidence, the new Mongo ID, we will re-evaluate if we want to lump the colon cancer or polyposis phenotype to this new curation. So again, here is the list. We found, we didn't find much evidence, and there is only limited evidence in the original curation. So we decide to make them as very low priority, and we will come back to deal with them after we finish high priority curations. Okay, so I would like to share our solution for the challenges. So here you can see we experienced some challenges, for example, new GCEP members or new things to the GCEP members, or like a limited GCEP capacity. So for the first part, we have new coordinator, that was me, and new curators and the new experts. And we also have so many things to GCEP members for example, lumping or splitting or GCI. So how we deal with that is we have introductory meetings for all of them, and I explained everything from our very beginning to like the last step of curation. And I also have a lot of follow-up emails to ask them, do you have any questions? If so, please feel free to reach out to me. And in addition, we have extra individual or small group meeting. So if they found some evidence, they are not confident to score them as like a certain score, we usually have this meeting and we discuss together to have a potential score or like potential plan. And next is about new but contradictory evidence or like a limited GCEP capacity. So for example, we only have one meeting a month and some discussion can be very long. So again, a follow-up email might be very important because you have to remind people, okay, so if you have new progress or and the next is again, the extra individual or small group meeting is also very helpful. So we can discuss those concerns together. And the other thing is the paired curation. So we paired a curator and a volunteer expert. So they meet before the group meeting so they can find something very important. And that might be, we need to point out in the group meeting. And they can also deal with something not, very straightforward, and they can present this topic very efficiently. And the last thing is the prioritization. So like we prioritize like high prioritization and low prioritization, and according to the importance of that curation. And finally, I would like to thank all current and past members in the GCEP, and also Mongo Omen and GCI VSEC group, and also gene curation working group, lumping, splitting working group, disease naming working group, and the homeostasis, thrombosis GCEPs. They help a lot during this recuration process. Yeah, that's the slide, and I'm happy to take any question.
Any questions for Sheng-An? If you're not able to talk for whatever situation you're in, also put it in the chat.
I think that's about like GCI didn't exist when, so I'm happy we have GCI now. So we have new technology, so to, entry those data more efficiently. Yeah.
Awesome. But if there's not Not any questions for Sheng-An. And I know he already mentioned that he'd be happy for you to contact him offline as well. But we might end it there for today. But thank you so much, Sheng-An, for that presentation. It was actually fascinating to hear the experience of a GCEP and your experience over time for a group that's been up and running for so long, and particularly all your experience around re-curating genes, which I think not many GCEPs will have that experience. So hopefully we can integrate what you've learned from that into our more general guidelines. So thank you again.
And feel free to feel free to reach out to me via email if you found some question after this presentation. So
wonderful. And just a reminder that our next meeting will be next Thursday on June 22nd, and we'll be at that presentation from Leslie Secker on behalf of the segregation linkage subgroup of the sequence variant interpretation working group. So we will see you all next Thursday, and thank you again, Sheng-An.
thank you. Bye-bye.